Hey guys, welcome to Let's Crack NEET PG, the YouTube channel for Unacademy, our NEET PG division. So today's class is another integrated class on neurotransmitters, right? So we'll discuss what are neurotransmitters and what are the various neurotransmitters, what are some important points related to these neurotransmitters, right? So the good thing about this lecture is that it is not just going to be a physiology lecture. I'm also going to put in biochemistry, right? Because you all know that there's a lot of biochemistry related to the catecholamines, especially, and the all the amines, right? So I'm going to discuss that biochemistry aspect also. And apart from that, there are some aspects in psychiatry related to these or there are some clinical aspects, some of them of psychiatry and some of them in general about Parkinson's, about Huntington's, about Alzheimer's. So all of these diseases, they are also correlated with neurotransmitters, right? So I will discuss it in an integrated way. I will try to make this lecture as interesting as possible for you guys. Okay. So I hope you guys are ready. I hope you guys are ready to start the class. Let me just put it on the channel. Okay. Right. So if you have any doubts, you can put it on the YouTube chat. I have the chat open here on my mobile phone, right? So you guys can put anything you want there. So let's begin the class. First of all, my name is Chetanya Mittal. I am currently pursuing my MBBS from All India Institute of Medical Sciences, AIMS, New Delhi, right? So I hope you've all already downloaded the Unacademy Learning app. In case you haven't, just go to the Play Store and download it. You will need to register with your mobile number and then select your goal as NEET PG. Use my referral code Chetanya10 to unlock a free plan for this goal, and you can access all the free content that is there on the Unacademy application, right? So all the top educators are teaching live on Unacademy. So there are plus courses, which are obviously paid courses. So these are paid courses. But at the same time, if you download the application, you can take advantage of the free of cost special classes, right? So today at 3:30 p.m., do join me for an MCQ marathon session, okay, for anatomy, and that is also going to be on special class. So just go to the description of this video and you will find the link for my Unacademy page. Okay. So there are batch courses, there are special tests, there are doubt clearing facilities on Unacademy, right? So you are getting a live classroom experience. You can attempt GTs and you can also clear your doubts. So all of this makes Unacademy a one-stop solution for your NEET PG preparation needs. We have recently started two batches on 7th April. One is for FMG. It is a test and analysis batch, 9 subject-wise tests, right? And one grand test. And then we have a 24-day MCQ marathon batch, right? So 19 subject-wise tests and 3 grand tests. So again, 22 tests taken up by the top educators in their respective fields. All right. So then these are the upcoming or the latest batches. You can see what are the batches that have been started for April. I've already told you about two. And these are some free of cost mock tests that you can attempt on the application. So Unacademy provides you flexibility like no other. We are giving you flexibility in your schedule, in your language, at the same time ensuring a live classroom experience by giving you robust chat feature and interactive polls. So you will see that how beautifully the polls are utilized when we have an MCQ marathon at 3:30 p.m. today. You can check out the link in the description for my Unacademy page and you can enroll for today's class. So there are two kinds of subscriptions. There is an Unacademy Plus and there is an Unacademy Iconic subscription. All right. In the Unacademy Plus subscription, you get access to the live classes, the batch courses, and in the Iconic subscription, you get everything that Prep Ladder has to offer: the video lectures, the question bank, the handwritten notes. All of that is included when you are taking a Prep Ladder, an Iconic subscription, apart from what is being offered with Unacademy. So in the Iconic subscription, you are essentially getting Unacademy plus Prep Ladder. All right. The 1 year subscription costs 55,000, but you can use my referral code Chetanya10 and you will be eligible for an additional 10% discount. All right. And that will cost you 49,500. Similarly, if you take the 2 year plan, it is for 77,000. However, you can again use my referral code and you will be eligible for a 10% off. Okay. So that is the comparison. And this is about the plus subscription. If you take the 1 month subscription, it is for 5,000. Again, at discount, it is at 4,500. The 1 year subscription is much cheaper at 25,000. So you are getting the 1 year plan for 25,000 and you can get it for 2 years at 36,000, right? So purchase it on an Android phone. This is how you apply the referral code. This is how you apply the credits. And that brings us to today's class. Okay, neurotransmitters and their clinical correlates. Okay, so and I've already told you that this class is an integrated session. This class is going to be an integrated session. What subjects have been integrated? Mainly physiology, biochemistry, and a touch of psychiatry. All right. Are you guys ready to begin? Can you guys say hello? Okay. So it's time to begin. First of all, let's talk about some very basic things. This is the synapse, right? So you are all aware of the synaptic transmission. I hope you've been studying this from class 11, at least from class 9, right? So how does a synapse work? So there is a presynaptic end, there is a synaptic cleft, and there is a postsynaptic end. So neurotransmitters, they are the messengers of synapse. They are the messengers at the level of the synapse. Okay. Hi, Elena. How are you? So neurotransmitters are the messengers at the level of the synapse. That is, they will take a message from the presynaptic end to the postsynaptic end. All right. So think of it in this way that there is a river in between and there are two banks. So neurotransmitter is the boat. Neurotransmitter is the boat. Now the condition is that so what are the steps that are happening? First of all, the neurotransmitter, it is synthesized and it is stored in what is called as a synaptic vesicle. All right. Then some stimulus comes, right? There is some signal that comes that opens voltage gated calcium channels that causes influx of calcium. So that is the signal, and when that signal occurs, there is a release of neurotransmitters into the synaptic cleft, right? And once they are released, they are going to go on the postsynaptic terminal where there are receptors. They will bind to these receptors and they will produce a signal at the postsynaptic terminal. Okay, so these three things that I have told you: synthesis and storage in the presynaptic terminal, release in the synaptic cleft upon receiving a signal, their release after receiving a signal, and their action on the postsynaptic terminal at the receptor. These three things constitute essential criteria for a substance to be called as a neurotransmitter. Okay, so when do you call a substance as a neurotransmitter? When it satisfies three criteria. When it satisfies three criteria. Now, what are those criteria? I've already told you. First, it must be synthesized in the presynaptic terminal and stored there in the synaptic vesicles. Second, it must be released upon signaling to the synaptic cleft. And third, it must act on the postsynaptic terminal. Okay. Hi, Tejasvi. Hi, Hit Mangukia, Hit Patel. Okay, so I think it's Mangukia Patel. Right. How are you? So the last criteria is that it must act on the postsynaptic receptors and it must produce a signal there. So that is the criteria for neurotransmitters. That is, it directly is the boat of the synapse. Now, what are neuromodulators? What are neuromodulators? Okay, so now that is a very essential question that your Ganong brings you to. Okay, in the first page itself, it differentiates between two kinds of biomolecules. One is neurotransmitters, the others are neuromodulators. Okay, so think of it in this way: neurotransmitters, they are boats. Neurotransmitters, they are going to be, wait. If you guys have any doubts in between, please feel free to interrupt me in the chat. Okay, it would still be better if you guys could interrupt me with audio. That would be amazing, but then that is not feasible, right? So neurotransmitters, they are going to be the boats. Okay, so they are boats that are helpful in crossing the synaptic cleft. They are boats. Okay, now what are neuromodulators? So neuromodulators, they are basically going to be this rudder behind. Okay, जो boat होती है उसके पीछे एक वो direction देने के लिए there is something attached, right? That gives direction. If you have seen a motorboat, there is a tail to that boat through which you can change the direction. Okay, so neuromodulators, they do not act directly on the nerve terminals, right? But what they do is They modify the effect of neurotransmitters. They will modify the effect of neurotransmitters. So that's the key difference. Neurotransmitters, they are acting directly. They are the boats, they are the messengers. But neuromodulators, they modify the effect of neurotransmitters. Understood? They are going to modify the effect of neurotransmitters. Okay. I think there might have been a slight disconnection in between. I hope it's fine now. Okay, understood. Difference between neurotransmitters and neuromodulators. Now, according to your Ganong, neurotransmitters, they are divided into various classes based on their type, right? So there are various classifications of neurotransmitters, and this one is the one given in Ganong, so we're going to stick to it. The first classification, this classification says that neurotransmitters, they can either be small molecules, or they can be large molecules, or they can be gases, right? So what can they be? They can either be small molecules, large molecules, or they can be gaseous. So these are the three ways to categorize neurotransmitters. All right. So small molecule neurotransmitters, for example, we can include glutamate. For example, you can have glutamate, or you can have, let's say, dopamine. Okay, so all of these have the capacity to act as neurotransmitters, and they are relatively small molecules. Okay, as opposed to large molecules. So three examples of large molecules are given in your genome, right? So what are those three examples given? One is substance P, second is enkephalin, and third is ABP. Third is ABP. Okay, so ABP is, I hope you all know, ABP is arginine vasopressin. ABP is arginine vasopressin. Any doubts so far? And gaseous neurotransmitters, there are two: nitric oxide and carbon monoxide. These are gaseous neurotransmitters. Any doubts so far? Any doubts in this classification? Please let me know. Okay, so if this much is enough, let's move to the second kind of classification that is there. That is mainly there for the small molecules, right? That is mainly there for the small molecules. And sometimes a question has been asked from this classification. Okay, so which was the first neurotransmitter that was discovered? The first neurotransmitter that was discovered, the first neurotransmitter that was discovered was acetylcholine. Okay, first neurotransmitter to be discovered was acetylcholine. Okay, first neurotransmitter to be discovered. So scientists said that since this is the first neurotransmitter to be discovered, let's put it into class 1. So class 1 neurotransmitter is acetylcholine, right? Class 1 neurotransmitter is acetylcholine. All right. Then second thing, then there are other types. Okay. So now this is not a complete classification, right? Because there are a lot of different ways in which you can classify neurotransmitters. Okay. But this is a broad classification. The second thing is biogenic amines. Biogenic means that they are being synthesized in the body, and amines, it refers to the amine group in them. So biogenic amines. We'll talk about what are the various biogenic amines. For example, dopamine, catecholamine, all of those. Then lastly, then third category, third class is amino acids. For example, they can be excitatory or inhibitory. Excitatory neurotransmitter, which is an amino acid, is glutamate. Inhibitory neurotransmitter, which is an amino acid, is glycine or GABA, right? So whatever you want to take it. And the last class of neurotransmitters is the relatively newly discovered ones, okay? So which includes a purine neurotransmitter, that is ATP, right? ATP is a purine neurotransmitter. Then nitric oxide, which is a gas neurotransmitter. So all of those miscellaneous neurotransmitters, they are included in class 4. Understood? This kind of a classification. In other ways, you can classify based on their specific structure. So right, you can have monoamines, where you will include certain substances. You can have trace amines, right? You can have peptides, like for example substance P is a peptide. You can have purines like ATP and adenosine, they are purines. Okay, you can have gaseous neurotransmitters. So all of these are different kinds of classifications which are going to work. Okay, so if you start with neurotransmitters, that is the basic in understanding what are the types of neurotransmitters. Understood? Now let's move to the now let's move to certain important points about various neurotransmitters. So now this is the part where I want to discuss. So this is initially I have discussed a little bit of physiology. No, I said adenosine. Aditya Agrawal, I said adenosine, not adenine. Adenine is a nitrogen base. I said adenosine. Okay. So that is sometimes a neurotransmitter. Okay. So I have discussed a little bit of physiology with you guys right now. ठीक है, अभी मैंने क्या किया है, थोड़ा सा तुम्हारे साथ physiology डिस्कस कर दिया. We'll come back to the physiology, we'll come back to the receptors, we'll come back to talking about the physiology. Okay, so physiology I have done a little bit. Okay, there is still some pending one. And now I will move to the biochemistry. Now I will move to the biochemistry portion, where we will discuss mainly the synthesis, the degradation, and all of that. Okay, so let's start with the first discovered neurotransmitter, that is again going to be acetylcholine. ठीक है, तो ये बात मैंने तुम्हारे दिमाग में डाल दी है, that first discovered neurotransmitter is acetylcholine. है ना, इसमें तो डाउट नहीं आएगा. तो इसकी बायोकेमिस्ट्री there is not too much to know about it. Okay, it's very relatively very simple. So acetyl CoA, you all are very familiar with this molecule, है ना, Krebs cycle में बहुत बार देखा हुआ है. What is acetyl CoA, right? It combines with a nitrogen compound called as choline. It combines with a nitrogen compound called as choline, right? And then there is an enzyme called as choline acetyl transferase. So now why is it called as choline acetyl transferase? Because it will transfer this choline group next to this acetyl group. Okay, या फिर उल्टा लगा लो, acetyl group to the choline group. Okay, so the essentially what will happen, this CoA will get removed, right? This acetyl will get rid of this coenzyme A, and it will take hold of choline. So it will lead to the synthesis of acetylcholine. All right, acetylcholine. So if you guys are liking this video, please do remember to hit the like button on this video. Okay, and one more small thing, we have a session at 3:30 p.m. which is an MCQ session on anatomy. Please remember to enroll for that particular class and attend it. We have Amazon gift vouchers for the winners. Okay, so let's start. Let's continue. So second thing is serotonin. Okay, so now this is the place where the actual biochemistry starts. ठीक है, अभी तक तो तुम्हें acetylcholine में तो क्या था, acetyl CoA और choline मिला दिया, acetylcholine. ठीक है, so serotonin synthesis is something that has a little bit of biochemistry. Okay, so the synthesis of serotonin, it starts from tryptophan. It starts from tryptophan, right? So what is tryptophan? Tryptophan is an aromatic amino acid. So you start from tryptophan and there are two steps in the formation. हमेशा याद रखना, जब भी ये synthesize करना है तो दो step होता है. पहला होता है hydroxylation, second होता है decarboxylation. So whenever you are going to be doing anything, remember we will first do hydroxylation and then we will do decarboxylation. Okay, so for hydroxylation you need something called as tetrahydrobiopterin. This is a cofactor. Okay, BH4, what I have written, tetrahydrobiopterin, right? And what is BH2? It is obviously dihydrobiopterin, right? I'm writing this here, it will come again. So there you have to remember what is BH4 and what is BH2. BH4 is tetrahydro, so 4 hydrogen, so therefore H4, and it is biopterin. So biopterin के लिए B लिखा है. So BH4. What is BH2? Dihydrobiopterin, right? So tetrahydrobiopterin gets converted to dihydrobiopterin. Tetrahydrobiopterin gets converted to dihydrobiopterin, and you have the utilization of oxygen. So this is an oxidation reaction, right? So this is an oxidation reaction. So in this, what will be the enzyme involved here that I have written as A? What is enzyme A? This will be called as tryptophan hydroxylase. Any doubts? Tryptophan hydroxylase ही कहेंगे कि कुछ और कहेंगे? Tryptophan hydroxylase, right? Now in the second step, I told you we will have decarboxylation. So now what will be the name of the enzyme here? Okay, so remember this was an aromatic amino acid. So now this enzyme is called as aromatic amino acid decarboxylase, right? And we will be using something called as PLP. What is the full form of PLP? PLP is pyridoxal phosphate. Okay, PLP is pyridoxal phosphate, and it is a cofactor. It is a cofactor of all decarboxylation reactions. It is vitamin B6. Okay, and carboxylic acid will be released, and you will get. So this was 5-hydroxytryptophan. Once it gets decarboxylated, it is called as 5-hydroxytryptamine. So you can see it is tryptamine. So it is a amine, tryptamine. So it is a 5-hydroxytryptamine, and that is another name for serotonin. So you will always see serotonin being represented as 5-HT. So now you know what is the meaning of 5-HT, 5-hydroxytryptamine. Okay. Parth Sharma, I am going to start my preparation for NEET. What should I suggest? Be more specific to that. Okay. NEET UG, NEET PG, what is your exact doubt? Okay. After that, let's move ahead and let's talk about the degradation. So once 5-HT is formed, it also has to be degraded. It also has to have a degradation. Okay? It has to be broken down. Otherwise, it will keep acting, and then the serotonin action will become overpowering. Okay? Excess of everything is bad. All right, simple principle. So how it is degraded? It is degraded by MAO. I'll tell you later on what is MAO, right? But for now, remember that 5-hydroxytryptamine, that is serotonin, it is degraded by an enzyme called as MAO. All right, and the product is 5-hydroxy 3-indole acetic acid. So it is written as 5-HIA. Okay, 5-HIAA, 5-hydroxy 3-indole acetic acid. ठीक है? इससे तुम्हें ऑक्सिन याद आ रहा है somehow? You guys remember what was auxin? Auxin में भी होता है ना indole acetic acid, 5-HIAA. So where was auxin first discovered? In human urine. Okay. So 5-hydroxyindole acetic acid, it is a metabolite of serotonin, and you can determine body serotonin levels by doing urine analysis of 5-HIAA. इसका urine analysis कर सकते हो तुम to determine the body serotonin levels. Okay, understood? Obviously इसका degradation product है. So if there is a high level of 5-HIAA, right, it means that the body serotonin levels are high. Okay, understood? Hmm. So there are some tumors which release large amount of serotonin. Which are those tumors? Do you have any idea about carcinoid tumors? Right? Carcinoid tumors are serotonin releasing tumors. Okay. And what are the other conditions? Okay. So अब देखो थोड़ा सा psychiatry correlate मैं यहाँ पे कर दूंगा. बाकी we have a separate section at the end. So if you want to correlate the levels of, right. So I think it should be back now. Okay. So that was some struggle that we went through. But anyways, thank you. You guys persisted. Right. So I was saying, what are the correlates when you are actually going to be doing 5-hydroxyindole acetic acid testing, right? So that is when you are going to suspect that the levels of serotonin are high. Okay. So it can be used in the diagnosis when we are suspecting a carcinoid tumor, first of all, because carcinoid tumors, they are serotonin secreting tumors. So if there is an excess of serotonin in your body because it is released by enterochromaffin cells and they secrete serotonin. Okay, so that shows that your body has an elevated levels of serotonin. Okay, elevated levels of serotonin. Okay, the second thing is a psychiatry correlate. Okay, that is a psychiatry correlate, and this is one of the few areas in psychiatry where you can have a laboratory investigation, and that is autism. That is autism or autism spectrum disorders. In that case also, it is said that serotonin is elevated in autism. All right, understood? Yes. So that is one thing. So that is what you need to know about the biochemistry of serotonin. ठीक है, तो biochemistry of serotonin is covered, है ना? Now let's move on to the biochemistry of amino acids, right? So amino acid neurotransmitters. Okay. So which class of neurotransmitters were they? Class 3 neurotransmitters. These were class 3 neurotransmitters. So one thing I haven't told you so far is when you talk about amino acid neurotransmitters, they can either be excitatory or they can be inhibitory, right? So which is the major excitatory neurotransmitter that you know of in the CNS? That is going to be glutamate. That is going to be glutamate. And when you talk about inhibitory, the major inhibitory one is GABA, and you also have glycine. So glycine. So glycine is the proper amino acid here. This is not a amino acid in your body, right? So, but otherwise, we are also considering it here for our purpose of study. So if you look at this, So how are they synthesized? How are they synthesized? So they are common. So since they are amino acids, they are common metabolites of your body's already existing processes. They are common metabolites of your body's already existing processes, right? So now why is that? Why is that the case? See here, alpha ketoglutarate acid, which is a metabolite of your Krebs cycle, which is part of your Krebs cycle. So in the Krebs cycle, there is an enzyme called as GABA T, right? So GABA transferase, and that will lead to the synthesis of glutamate. So that is the first pathway of glutamate synthesis. All right. Inside the nerve terminal, there is a second pathway where there is a amino acid called as glutamine, on which you have the action of an enzyme called as glutaminase, and that will lead to the synthesis of glutamate in the nerve terminal. Okay. So this is how the excitatory neurotransmitter glutamate will be synthesized. Excitatory neurotransmitter glutamate will be synthesized in this fashion. All right. Now, if you want to synthesize, now guys, if you want to synthesize GABA, synthesis of GABA carried out. Okay, so there is an enzyme GAD, that is glutamate decarboxylase, right? So carboxylic acid is going CO2 is going to be released. Whenever we are doing decarboxylation, we are releasing carbon dioxide. We are releasing carbon dioxide. So glutamate decarboxylase is the name of the enzyme. Glutamate decarboxylase is the name of the enzyme, and the product is GABA, and the product is GABA. The product is GABA. Anyone has any doubts? Anyone has any doubts? Any doubts? Okay. So that is about amino acids. That is about the basics of amino acids. All right. Now, when you go ahead, when you go ahead, let's talk about catecholamines. Let's talk about catecholamines. Let's talk about catecholamines. Okay. So catecholamines is the actually intensive place regarding biochemistry. It's the actual intensive thing where you will have a lot of biochemistry, right? This is the place where you will have a lot of biochemistry. Okay, biochemistry. Okay, so what is the condition here? What is the condition here? Catecholamines. Catecholamines. You start from tyrosine. You start from an amino acid tyrosine. Is tyrosine an aromatic amino acid? Yes, very much like tryptophan. Very similar to serotonin. देखना सेरोटोनिन कैसे सिंथेसाइज होता है, और जब सेरोटोनिन से कंपेयर करोगे तो तुम्हें डेजा वू होगा. क्या होगा? डेजा वू. देखो सेरोटोनिन का देखो एक बार. देखो सेरोटोनिन, यू स्टार्टेड विद ट्रिप्टोफैन, यू डिड हाइड्रोक्सिलेशन, यू डिड डीकार्बोक्सिलेशन. ओके, अब डेजा वू लो. देखो यू स्टार्टेड फ्रॉम टायरोसिन, यू डिड हाइड्रोक्सिलेशन एंड यू डिड decarboxylation. All right, so it's the same sequence. You start from tyrosine, okay, you go to dopa, and then you finally go to dopamine. कौन है? So डेजा vu हुआ. देखो टायरोसिन से you will start. From tyrosine you will go to dopa. From tyrosine you will go to dopa. The second metabolite is dopa, right? After tyrosine you move to dopa, and from dopa you go to dopamine. Okay, tyrosine, dopa, dopamine है ना. ये sequence बहुत important. Again first thing that we will do will be hydroxylation. And hydroxylation के लिए what did I use there? टेट्राहाइड्रोबायोप्टेरिन. टेट्राहाइड्रोबायोप्टेरिन को मैंने डाइहाइड्रोबायोप्टेरिन में कन्वर्ट किया, ऑक्सीजन से पानी निकाला, और क्या किया यहाँ पे एंजाइम कौन सा आया? टायरोसिन बीटा हाइड्रोक्सिलेज. ठीक है, थोड़ा फर्क है, ये बीटा वर्ड थोड़ा देख लेना है ना. टायरोसिन हाइड्रोक्सिलेज, टायरोसिन हाइड्रोक्सिलेज. ओके, हाँ बीटा मत लिखना भी. टायरोसिन हाइड्रोक्सिलेज. when you go from dopa to dopamine, when you go from dopa to dopamine, what are you doing? Decarboxylation. You are doing decarboxylation. Okay, so when you are doing decarboxylation you are using a coenzyme. And what was the name of that cofactor that you always have to use? Pyridoxal phosphate, which is a form of vitamin B6. And what is the name of the enzyme here? Same enzyme as we were using previously, aromatic amino acid decarboxylase. Aromatic amino acid decarboxylase. Understood? Are we with this lecture? And I know that kind of spoils the user experience as well. Wait, let me just check. Right. Hey guys, so welcome to Let's Crack NEET PG. Yeah. So the point that I was emphasizing on before, if you are watching this video in continuity, hopefully you guys are. The point that I was emphasizing on was the fact that dopamine gets converted to norepinephrine. So dopamine leads to other catecholamines to be synthesized. So dopamine, you have an enzyme here, and the enzyme here is dopamine beta hydroxylase. So what I've written here as C is dopamine beta hydroxylase, right? Dopamine beta hydroxylase, and that actually converts dopamine to norepinephrine. And here you will utilize vitamin C, so you have it in the form of ascorbate. And again you're going to do the same thing, you're going to hydroxylate in the presence of copper, right? So you have copper here, and that will lead to the formation of norepinephrine. Now, under conditions of stress in your body, there is a response in your body with the help of glucocorticoids, right? So, what do glucocorticoids do? They activate an enzyme called as PNMT, that is phenyl ethanolamine methyl transferase. Okay, so forget about everything. Just think that it is a methyl transferase. So, what it does is there is a compound called S-adenosyl methionine, right? S-adenosyl methionine. So what it does is it transfers the methyl group from SAM to norepinephrine. So norepinephrine, when you add the methyl group, so you can write norepinephrine CH3. So this is called as epinephrine. So epinephrine is nothing but methylated norepinephrine. So that you will get, and SAM will get rid of a methyl group, and that becomes S-adenosyl homocysteine. So SAM gets converted to SAHA, and from norepinephrine you get epinephrine, and that is done under the influence of glucocorticoids, right? So I wanted to finish the biochemistry part here, right? So to sum up the degradation, have a look here. Have a look here. Dopamine, how does it get degraded? There is an enzyme called as monoamine oxidase, right? There is an enzyme called as monoamine oxidase, and when that acts, there is an intermediate that is formed. Okay, this intermediate is not important. Okay, so what you have to remember, I will tell you what you have to remember, that there are two enzymes. Okay, one enzyme is called as MAO, or that is monoamine oxidase. Dopamine is a monoamine, so you will have the action of monoamine oxidase, that is MAO. And there is another enzyme called as catechol O methyl transferase, or COMT. Okay, so now these two enzymes, they can act in any order. Okay, MAO can act first and then COMT can act, right? COMT, or COMT can act first and then MAO can act. In whatever order they act, it does not matter. These intermediates are not important for you. What you have to remember is that when you have MAO, COMT, in whatever order, okay, in whatever order, the final product from dopamine is homovanillic acid. That is important for you guys to know. What is the metabolite formed after the degradation of dopamine? That is going to be homovanillic acid. Understood? Is that part clear? No doubts in that part? Okay. So dopamine gets degraded to homovanillic acid. Now, after the formation of homovanillic acid, the second thing that you have to understand is the same thing for epinephrine and norepinephrine. Okay, so epinephrine and norepinephrine, both of them can get acted upon by MAO plus. So what you have to remember is MAO only. Okay, so monoamine oxidase. So both of them the same thing. Either they can have the action of MAO first and then COMT, or they can first have the action of COMT and then MAO. And the product that will be there will be vanillyl mandelic acid. So what was the product from dopamine? Homovanillic acid. And what is the product here? Vanillyl mandelic acid. Okay, so if MAO acts first, it goes via dihydroxy mandelic acid. So you can see here, if I start from epinephrine, MAO first, COMT later, you reach at vinyl mandelic acid. On epinephrine, COMT first you get metanephrine, then MAO, you again get vinyl mandelic acid. If you start from norepinephrine, MAO acts, you get dihydroxy mandelic acid, then COMT acts, you get vinyl mandelic acid. On norepinephrine, first COMT acts, you get normetanephrine, right? Here you are getting metanephrine, here you are getting normetanephrine. And then MAO acts, you get vinyl mandelic acid. Okay, so this table, it should be very clear in your mind. This whole image, it should be etched in your mind. Okay, so I'll tell you what are the important things to remember here. First of all, the enzymes monoamine oxidase and catechol O-methyl transferase, that is called as COMT. That is called as COMT. That is important. So you can remember, if COMT acts, the product from epinephrine is metanephrine, the product from norepinephrine is normetanephrine. And finally, what you will get will be vanillyl mandelic acid. Vanillyl mandelic acid, that is the product from catecholamines. What is the product from dopamine? Homovanillic acid. What is the product from catecholamines like epinephrine, norepinephrine? Vanillyl mandelic acid. Okay, is that clear? Right. So I have finished up some of the physiology part and some of the biochemistry part, right? So today we had some issues because of which we could not finish the lecture. I will do a part 2 of this lecture and we will try to finish up the remaining physiology. Okay. So what is still left to be discussed are the receptors, the various receptors of the various amino acids, of the various neurotransmitters. And at last we have to discuss the psychiatric correlates. Okay. When is serotonin increased? When is norepinephrine increased, and all of that. Okay, so we will discuss that in detail in part 2 of this lecture. Meanwhile, I really thank you for your patience, and do remember to hit the like button for this video and subscribe to the channel Let's Crack NEET PG, so that you know when is part 2 of this lecture coming. And you can subscribe to the Telegram channel. The link is in the description, @ Let's Crack NEET PG. That is the Telegram channel. All right, and do remember to hit the bell icon for notifications. And my plus referral code is Chetanya10. In case you wish to unlock a free plan on Unacademy or take a subscription, you will get a